Hi everybody, my name's Mason. I'm going over the antibody structure. So this is my antibody. This is the one I made with Play-Doh from Walmart, and it kind of models the picture to the right and some others that I found through research. Antibody overview. Antibodies fight pathogens and viruses introduced to the body. It protects against intracellular infection by recognizing different pathogens and viruses. It also facilitates the process of elimination of pathogens by initiating phagocytosis. Phagocytosis is the ingestion of a pathogen by the way of phagocytes secreted into the blood or mucus and travel the body through these mediums. That is, of course, how antibodies attack infectious sites and summon other cells or other antibodies to the site to attack these pathogens. Heavy chain. The heavy chain provides the most structural support in the cell. It's the largest portion of the antibody. Consists of two portions. The constant region consists of the M, G, A, E, and D stems, depending on the antibody, and the variable region, composed of a single domain, has an antigen binding site near the end of this part of the heavy chain. 5 classes of isotopes of the heavy chain: IgM, IgD, IgG, IgA, and the IgE. You can see the green right here is the heavy chain. The light chain, smaller portion of the polypeptides that runs parallel to the heavy chain, bonded to the heavy chain by disulfide bonds. Like the heavy chain, the light chain has a variable and a constant domain. This aids in offering an antigen bind site, and it is a little over 200 amino acids long. You can see here it is with the arrows pointing to the red. That is the light chain. The paratope, region of the body that can bind to the epitope, the result of the bind of both types of the chain, the heavy and the light chain. This is known as an antigen binding site located at the distal end of the heavy and light chains. You can see I pointed to it here, but it's the CLO at that far distal end of the heavy and light chain. The disulfide bond, this forms a link between the heavy and the light chains, stabilizes the antibody structure as a whole, acts as a bridge from the light to the heavy chain. Disulfide bonds are also present in proteins. It allows for different structural changes to the antibody. In turn, the function of the type of antibody it has created through the different bonding sites. You can see it correlates here to here. The hinge serves as the link between the different types of heavy chains, allows for more flexibility within the structure and function of a heavy chain, aids in complement binding in an antibody, can change the binding complement due to the flexibility in the hinge. This can change the function of said hinge. You can see it, skinny portion right here. These are my references. That is a quick overview of the antibody structures and a quick look at the function of said antibodies. Thank you, and have a good night.